All right, we're on concept 11.3. This is part 2, and now we get into the details of transduction, and these are the more complicated aspects of this lecture. Transduction is a series of cascades. Basically, one molecule affects another reaction and triggers another reaction from one to the other, all the way until you get to the end product, the end of the pathway. So these multi-step pathways, they're ideal, and every reaction is regulated by an enzyme, and this enables you to control that, to regulate it more closely, because you can amplify the signal, you can turn it on more so, and you can also shut it off should you want to, because every step is regulated by an enzyme. And those enzymes can undergo conformational changes to be turned on and off, of course. Many of these different cascades are phosphorylation cascades, where one molecule is phosphorylating another, which is phosphorylating another, and so on. The enzymes that are kinases are very well known to do this. They commonly add a phosphate to the next one in line and then activate that enzyme, and so on. And then to cleave or remove that phosphate group from those kinases, there's a different enzyme, and we call those phosphatase enzymes, and they remove the phosphate groups. So here's what a phosphorylation cascade looks like. We bind a ligand, receptor triggers or activates this molecule, followed by this protein kinase being activated into an active form. And now you see active protein kinase 1 is going to phosphorylate kinase 2 to activate it. Here it's phosphorylated. And then kinase 2 will activate kinase 3. You notice it's phosphorylated with a new ATP and so on, all the way down until we get to our cellular response. That cellular response can be triggered through a series of second messengers, which are non-protein small molecules that are typically water-soluble or they're ions. One example of a very common one is cyclic AMP, or CAMP, C-A-M-P, and it comes from ATP. Here's ATP, and the enzyme adenyl cyclase removes the phosphate groups and cyclizes, or the phosphodiesterase then cyclizes to make, the ATP gets cyclized by adenyl cyclase. Here you see cyclic AMP. And then if you want to convert that to just AMP, you would use a phosphodiesterase. But this is not important. This is the target molecule that we want, cyclic AMP. It's a second messenger. So here we have an example of a G protein, which triggers the formation of cyclic AMP. G protein is activated with GTP, binds to adenyl cyclase, turns it on to convert ATP into cyclic AMP. And that cyclic AMP now triggers a phosphorylation cascade to have a cellular response. So you see a lot of mixing of these different types of pathways here. Cyclic AMP is not the only second messenger. There's others, of course, and this includes calcium, for example. It's actually more common than cyclic AMP. It acts as a second messenger. Here we see calcium levels inside the cell. Calcium is commonly stored in the ER, and also you see in the matrix of the mitochondrion. It's pumped in there to intentionally keep calcium levels low in the cytosol and very high inside these storage organelles for release later at a certain point whenever you want. You don't want calcium floating around all the time in high levels because it is a second messenger and it turns on metabolic reactions. Other second messengers include IP3, inositol triphosphate, and DAG, diacylglycerol, and those actually they work with calcium to trigger the release of calcium from the organelles. So here again we see G protein receptor, G protein gets activated with GTP, causes a conformational change here. In this case, phospholipase C is broken down into two different products, IP3 being the important one for our discussion. This is the second messenger. You also see DAG, but we won't talk about that one. That's a different pathway. IP3 binds to this ion channel on the ER and triggers the release of calcium from the ER out into the cytosol, which will then trigger a cellular response. So here's another example of a cytoplasmic response due to cyclic AMP and the kinase pathway you'll see, a phosphorylation cascade. Here we have epinephrine. Epinephrine is a hormone, also known as adrenaline. It triggers basically like a boost in energy, increases heart rate, increases respiration rate, you burn more sugar. So when we burn more sugar, we need more sugar in our bloodstream. So the G protein gets activated. which activates adenyl cyclase, and that triggers ATP to become cyclic AMP, which triggers this phosphorylation cascade from protein kinase A to another kinase, which in the end activates this enzyme, which will convert glycogen into glucose 1-phosphate. G1P, or glucose 1-phosphate, is basically one of the intermediates in glycolysis. So glycogen is essentially freeing up glucose molecules to use in glycolysis and respiration for energy. Glycogen is the storage molecule for glucose in liver and in muscle cells. So this is a real-life scenario of how you release glucose under stressful times or when you need more energy. This is showing you again kind of how that would work. Here's your phosphorylation cascade, triggering this transcription factor to turn on this gene, and the gene gets converted into messenger RNA and then into a protein. A little bit about fine-tuning of the response, so amplifying and turning down the response of these different types of cascades. There's different kinds of combinations, but all these different cascades are very highly specific for both the signals that it detects, the ligand that it binds to, and also the response that it's going to end up carrying out. So here's an example where we have very specific responses. Here, signal molecule ligand binds to this receptor, causes response 1. The same ligand binds to a different receptor, causes different responses. All right? Then we see here the same ligand binding to a different receptor, we have a different response. But if I have different ligands binding to different receptors here, sometimes these intermediates in the cascades will crosstalk, as we say, or interact with one another, creating an entirely new or different response. Sometimes it's not even a different response, it's just an increase or an amplification of response 5 or something like that, but oftentimes it's just an entirely new response. And you can see that there with crosstalk. Here's some text that I pasted from the web on crosstalk, just to give you a better idea of how it's explained. You can go ahead and read that on your own. I'm not going to go through reading it. But it's basically talking about crosstalk through second messengers, and it can be both beneficial and more difficult on the cell. Here's more on crosstalk and specific to cyclic AMP and calcium, how they interact with one another. And a real-life example here, AVP is also sometimes known as ADH, Antidiuretic Hormone. It triggers, it's the opposite of antidiuretic hormone. This is called vasopressin, is another name for AVP. And it triggers fluid secretion in the mammalian kidneys, but at the same time you have these prostaglandins that are opposing fluid secretion. They attenuate or minimize the intensity of the fluid secretion, and thus the overall response to the hormone is due to these multiple different transduction pathways that are kind of fighting each other to make for the ideal perfect response. Sometimes these proteins, these receptors in the membrane, incorporate the use of a scaffolding protein to bring all these kinases in very close proximity to one another, so it's very easy to phosphorylate and activate one to the other, enhancing or boosting the signal transduction efficiency. So this is a scaffolding protein. And the signal will end when the ligand binds or is removed from the receptor. Then the transduction pathway basically gets turned off. And that's the end.